to this Oregon Wild Wednesday webcast. We'll go ahead and get started. Thanks for joining us tonight. I am Chandra Legui. I'm Oregon Wild's Senior Conservation Advocate in our Eugene office, though it looks like I'm sitting in a wildflower meadow because that's where I'd rather be. I will be your guide through this webcast tonight. And if anyone here is new to these webcasts and to Oregon Wild, I'll just give you a brief orientation before we begin. So Oregon Wild works to protect and restore the wildlands, wildlife, and waters in our state. We've been advocating for public lands, wildlife and their habitat, wild and scenic rivers, and old growth forests that grow across the state since 1974. So we're pleased to be presenting these Oregon Wild webcasts so that folks at home can experience some amazing learning opportunities about our natural world. And tonight we are excited to be talking about a group of amazing fellow creatures, amphibians. So before we get into it, a few housekeeping items. For folks who may have bought raffle tickets when they registered for the webcast, we will do that drawing tomorrow and then email the winners. We have a couple of pieces of Oregon Wild swag, and I think maybe one of my Oregon's Ancient Forest hiking guides. And you can get raffle tickets where you signed up for the webcast on the Oregon Wild website. Just go back to that event and you can basically like register again, but buy raffle tickets. We do have some upcoming events. One of those you'll hear more about later tonight, which is our Wildlife Day of Action next Wednesday. So not a webcast, but something in person and very fun that y'all can do. And then the week after that, we have a program called Rooted in Place, Indigenous and Local Artists in the Blue Mountains, which I think is going to be amazing. So you can find those and registration on our website. Finally, as this webcast goes on, if you have any questions that come up, you can use the little Q&A bubble in your Zoom interface, and we will be answering questions at the very end of the program. We'll try to leave about 10 minutes for Q&A at the end. So try to put any questions that come up in the Q&A as we go along so that we don't get a million at the very end. And then before we go further, I do just want to recognize that the lands that we're going to be talking about today and are home to people since time immemorial. And this map just shows the traditional homelands of various tribes that call this place home. And the history of tribes in Oregon is really complex and nuanced and includes a lot of colonial legacies and wrongdoings like forced removal. And those have had long-lasting and current impacts to both people, landscapes, and the wildlife that we all share it with. So we believe that we all have a responsibility to not only acknowledge Indigenous people on the land, but also to do meaningful work to respect and uplift tribal sovereignty. All right, so let me introduce our fantastic speakers for tonight. So Dede Olson is a retired ecologist. She has worked for decades for the U.S. Forest Service at the Pacific Northwest Research Station in Corvallis and has also been teaching at Oregon State University. She's got a great presentation for us tonight about how amphibians have evolved as our landscape has changed at a geologic level. Big picture, as well as some of the challenges that they've overcome to succeed and those that they're facing today. And then we will also be hearing from Suze Wehr. Suze is a fantastic artist, as you will see in her presentation, an environmental activist who's currently working in communications and advocacy for the Western Environmental Law Center. She's a freelance illustrator and writer and recently released a guidebook for youth audiences called Amphibian, about amphibians, stories about Oregon's amphibians. And she's going to be filling us in tonight on a historic opportunity to fund wildlife conservation, including for amphibians in Oregon. So without further ado, I will stop sharing and invite Dede to share her screen and start the presentation.
Okay, let's see. Can you see?
Yep, looks and sounds great.
Excellent. Thank you for the introduction. I'm Dede Olson. I'm now an emeritus scientist with the U.S. Forest Service. I work out of Corvallis, Oregon, and I'm going to talk about tying together the geologic history, the advancing genetics that have informed our amphibian biodiversity over the last several decades, and how these tie together to inform conservation of amphibians. And I'd like to say that Chris Cousins really inspired this work. He is a PhD candidate at Oregon State University right now, and he will be finishing this year, 2026. And some of his work is represented at the very end of this talk to show how these different themes really can be pulled together. So let's see if I can figure out how to get the slides to forward now. There we go. So we're going to start with some geologic history, and this image of Pangaea might be familiar to many of you. It shows the supercontinent when our existing continents today were together 300 million years ago. And then continental drift started to happen, and the continents as we know them today drifted apart from one another. So here's a little of the geologic history that is very much tied to the species history on Earth. And at first, this was revealed with fossil evidence. And it was clear as fossils were being found on different continents today that were very similar to each other. And people could put the puzzle pieces of those continents together to show what had been at some distant past time. And today we can do the same thing, except not with fossil evidence, but with genetic evidence, because there's a genetic signature, a genetic patterns track species and their divergence over time with a DNA record. And there's essentially a molecular clock of genetic divergence of species over time. And as the last few decades have rolled past, those genetic advances have gotten more and more sophisticated. And we're learning more and more about how species and geology are very intimately tied. So let me do some basic concepts first. So we covered a little bit with geology. Let's cover a little bit about what is a species. And when I went to school in the 1970s, we had the biological species concept. And many of you are probably familiar with that organisms with common characteristics that can interbreed and produce fertile offspring are members of the same species. So the example I've given here is that horses are a species and zebras are a species. Although they can interbreed, their offspring are not fertile, they're sterile. So this is the definition of a species. They cannot produce fertile offspring. Horse is not a zebra, they're different species. Today, we have a more sophisticated view of what a species is, and it's based on the phylogenetic species concept. An evolutionary lineage has evolved separately from other taxa, and so we can look at the genes and how the genetic structure of these different organisms are in fact different, and also their morphology may be different. So the phylogenetic species concept is similar to, the horse and zebra one I just gave, except it looks at the finer spatial resolution of what makes up a species, the DNA. And the example here that I've given with salamanders, there's 3 species represented here, A, B, and C. These are Encetina salamanders, by the way. They live in woodland and forest habitats in the Pacific Northwest and into California as well. Encetina salamanders A, B, and C are different species. The lines you can see in that image give a clue of what is going on with the phylogenetics, the genetic structure. The line at the very bottom of the image that goes up to A, that is the basal lineage of this group. It is the ancestral lineage. So species A is derived from the basal or ancestor of all three of these species. Species B and C derived from A's ancestor. So B and C are derived species. Species C is somewhat unique. Um, you can see there's a map of California, and there's a ring around the Central Valley of California with different colors. The colors are representing different populations of the Ensatina salamander species in California. There are different populations around that ring. Along that circle, along that ring, the adjacent populations can interbreed. However, when you go from like the southwestern coastal part of California up and around and down through the Sierra Nevada mountains, those ends cannot interbreed. This is called a ring species, and it's just an example of unique populations along that ring. And then this whole diagram represents how different species develop. So A, B, and C are different species. They have different genetic makeups. And in this example, you can see that they look different. They have different color morphology. Oftentimes, there's other morphological characteristic, head shape, arm and leg length, tail length, these other things that may differ to define different species. So what causes species to diverge? And there's a separation often between different populations and then time. And over time, there are either adaptations that change the genetics and morphology of those organisms, or there's drift, just random mutations that may happen. And some of the reasons why animals and plants and other species get separated comes back to geologic forces and abiotic forces more generally. So I'm going to talk about mostly the geologic forces in the examples I give today for the Pacific Northwest. And I've already mentioned plate tectonics and continental drift, but we're going to have some other examples of mountain building, landslides, volcanism. And as mountains are built up, then there can be atmospheric changes as a result of that in terms of rain shadows. But atmospheric events can be, atmospheric changes can occur through volcanic emissions or greenhouse gases and other processes as well. And there will be some examples I talk about of water and how hydrology may separate populations. And already with that example of the ring species in California, the Ensatina, what happened there, that was during the Pleistocene era, and there was a lake in the middle of California. So that was a hydrology example. Lake Corcoran occurred during the Pleistocene in Central California, and that led to that ring species occurring around the lake. So we're going to skip now to just some foundations of conservation. And this history comes from my own personal history. When I took my first biology class in college, the instructor of that class happened to be Dr. Michael Soule. And he had a strong conservation ethic. And some people think of him as being the father of conservation biology. When I was at school at UC San Diego, where he was teaching, he put together a conservation biology conference, and it was the very first international conservation biology conference. And I was fortunate enough to help with that conference, mostly by doing undergraduate grunt work, which meant driving people to and from the airport and that sort of thing. But I met amazing people at that time because of it. There were two main objectives of this conference. One was to focus on nature conservation. And secondly, a recognition that to conserve biodiversity, we needed conservation genetics. And genetics was very different at that time. I really associate genetic studies to gel electrophoresis, a technique that was common at that time, and working with fruit flies, really. Although certainly there were genetic studies going on with a lot of different organisms. But even at that time, there was a broad recognition that there was a biodiversity crisis ongoing, and we needed to identify species before they became extinct. So back then, in the 1970s, there were about 1 million species known. And today, we've doubled that. So we have quite a bit of knowledge gain in these decades that have passed. And there's about 2.3 to 2.5 million species known today. However, there is estimated that there's over 8 million species on Earth, and not exactly knowing how many, but that's a number that we often read in the literature. So in this knowledge advancement of how many species there are on Earth, there has been another recognition of how many species are at risk of going extinct. And for vertebrates, which are those species that have backbones, including mammals, reptiles, birds, fish, and amphibians. Amphibians, unfortunately, lead the way and have the most species that are estimated to be at the risk of extinction. 41% of the species on Earth, of amphibian species on Earth, are at risk. And we don't really know exactly how many amphibians are here yet. Like that previous slide, we're in that knowledge discovery as we go along. But the first counts of amphibians happened in about 1985, and that count was about 4,000 species. And today we have a little over double that. And so people definitely have been at work trying to identify what the biodiversity is for the vertebrates that are tied to water, these amphibians, as we go along. So I'm going to focus my my talk now on amphibians, and we're going to get to the Pacific Northwest and start pulling these different themes together of amphibians, conservation, genetics, and geology. So just for some context, at the world scale, there are about 8,000 frogs and toads in the world, about 800 salamanders and newts, quite a few less. And then there's a third group of amphibian called caecilians. They live underground. They don't have legs. They look a little bit like worms, and they occur in tropical areas of South America, Africa, and Southeast Asia. But you can see that frogs and toads really dominate amphibians at the world scale. When we get to the United States, we find that it is a world salamander biodiversity hotspot. This is something unique to the United States. There's more salamanders and newts in the United States than there are frogs. And this is even more extreme when we get to the Pacific Northwest of North America, where there are twice as many salamanders and newts than there are frogs. We have about 50 species here, and they belong to 3 different general assemblages. So these pictures on the leftmost screen, you see a Cascade torrent salamander. This is a stream breeding amphibian. It's a salamander that breeds in streams, and then it can go up to land as after metamorphosis as an adult. The Van Dyke salamander is a member of the plethodontid salamanders. These are called woodland salamanders. Most do not have a need to go to standing or flowing water at any time in their life. However, this particular one, Van Dyke's salamander, does seem to be associated with seeps. So it does have an aquatic tendency, but many of the plethodontids do, they live in talus or rocky substrates, or perhaps in downwood. They do not necessarily have to go to any sort of water. The rightmost is a cascade frog, and that is an example of a pond breeding amphibian. So it is not necessarily associated with flowing water, but ponded water. So I started working with amphibians in 1982, and that's why I put 82 on this map and comparison of how the number of species have really grown in the Pacific Northwest in my experience. So between 1982 and 2026, these two groups have expanded quite a bit. Um, we have the plethodonid salamanders. These are the terrestrial salamanders, and the yellow, um, no, not the yellow yet. If you just look at 1982, you can see what species we had recognized at that time. And then if you go to the next column over 2026, you can see for each row, there are more species recognized. And this happened through genetic analyses. So Aeneades varius is the clouded salamander. And today we recognize two types of clouded salamanders. There's varius and vagrans, and so on down the line. On the right, the headwater stream amphibians, so the stream breeders, also have expanded in numbers over this time frame in the last several decades. And the giant salamanders, Dicamptodon, at one time, there was one of those. Now we recognize four. Rhyocotryton are the torrent salamanders. There used to be one. Now we recognize four species. And Ascaphus are the tailed frogs. There was one, and now we recognize two of those. So the yellow highlighting are examples of those species that we have some conservation concerns about. As the species have been recognized, their geographic ranges become smaller and smaller. And some of these conservation concerns are because of small ranges. Others may be concerned because we just have limited information about them, or there may be threats that are of concern for them. So in addition to species level biodiversity, there is population level biodiversity as well. And here are just a few examples with the plethodonid, those woodland salamanders. Underneath that line, as you go down, there are clades or distinct populations now recognized for some of these plethodonid salamanders. So for example, Plethodon stormi, right below the yellow line, that's the Siskiyou Mountain salamander. There are two distinct populations now recognized for that one. And the next one down, the Van Dyke's salamander, Plethodon vandykii, there are two different populations recognized for that, etc. And today, in some cases, a distinct population segment or a clade might be a unit to categorize at-risk populations for conservation. So species level conservation matters are, of course, a concern, but now in some cases, population level distinctions may also be used for conservation. So going back to this idea that we're going to put the puzzle pieces of geology together with amphibian biodiversity for the Northwest, let's start with that Pangaea example again. So just for context, we think amphibians emerged on Earth from that primordial soup so many million years ago in the range of 360 to 370 million years ago. That's when amphibians emerged. Then the supercontinent of Pangaea split only about 100 million years later, a blink of the eye, right? So at that time, at the time of Pangaea, there were amphibians on the land and in the waters. And there is one group of amphibians in the Pacific Northwest that has ties to Pangaea. It is the most primordial amphibian on Earth today. We happen to have it here in our area. It's one of our iconic species, and maybe you've heard of it. It's the tailed frog, species Ascaphus. There are two of these. They are the most primitive, the most ancestral amphibian on Earth. It is a remnant of amphibians since Pangaea. The closest relative of the tailed frogs in the Pacific Northwest occur in New Zealand. So look at the map. New Zealand's pretty far. They did not swim there. They, at one time, were a contiguous species across Pangaea. And as the continents broke up, they were separated. And over time, there has been either adaptation or genetic drift, just random drift of the genome of these groups. So here's a picture of the New Zealand species that was most recently put into a different genus. It had previously been in the same genus as ours here in the Northwest, Escaphus. And you can see my citation of this recognition of the divergence of these groups is in sync with what we think. So Pangaea was splitting in about 200 million years ago, and the molecular clock of the genetic work that the scientists have done suggests that a divergence of the New Zealand and the Pacific Northwest forms of tailed frogs, they separated, they diverged about 150 million years ago. So things are matching up here. There's one other species from the larger Northwest that has a Pangaea story, and that is the Hydromantes, Shasta salamander. If you look at the map on the left, you can see an arrow going to a dot. That is essentially Lake Shasta, Shasta Lake. And right there in the limestone rock is where the Shasta salamander occurs. It has a couple sister species, different species. When they're closely related, we call them sister species in California. But because I want to emphasize the Pangaea story, they do have a sister species in an unusual place. Let's see if I can get my mouse to go to the right place. There we go. And it's Italy. So of all places, well, there's a lot of limestone in the Eastern European area, and they are busy discovering new species there. And so these limestone or cave salamanders in Italy are very closely related to Hydromantes. And the only way they can get there, right, is this Pangaea story. At one time, these continents were connected, and the separation, the divergence of them over time occurred with the drift. So now I'd like to focus more on the Pacific Northwest and put several mountain ranges into the picture and what has happened to amphibian biodiversity due to these mountain ranges. And the first one is the Rocky Mountains. The Rocky Mountains formed about 40 to 80 million years ago. And this effectively separated amphibians in the East and the West. There are 50 species in the Pacific Northwest of amphibians. Only 5 of those 50 species have ranges that extend east of the Rockies. So 45 are endemic to the Northwest. That's 90% are endemic to the Northwest. So we have unique amphibian fauna here. Thank you, Rocky Mountains. That means the Rocky Mountains have been a barrier to dispersal for most of the species. And of course, animals are associated with habitats and microhabitats, and that will become part of the picture as we go on as well. So another mountain range, the Cascade Range, the modern Cascade Range at least, its uplift is tied to about 10 million years ago. And as it rose, it created a rain shadow in the Columbia Plateau as well as the high desert of Oregon. And we put that together with the northern Rockies, which go through Idaho. And we kind of have this like Oreo cookie sort of thing happening, where we have the northern Rockies being moist coniferous forest habitat, the Cascade Range being forest habitat and moist coniferous forest on the west side, and a dry place in the middle. And there are 3 pairs of species that show this divergence of the geography, the geology of the area in their genetics. They are sister species again, different species, but very closely aligned. The top pair In Idaho is the Coeur d'Alene salamander that occurs in the Rocky Mountains, essentially, of Idaho and into Canada. And then the Van Dyke salamander that occurs in Washington state. And you can see three populations of Van Dykes in Washington state. So these are very similar species that are split, we think, likely by the Cascade Range growing and the dry plateau intervening that is not habitat for the species in the middle. Similarly, we have the Rocky Mountain giant salamander occurring in the Rocky Mountains and the coastal giant salamanders occurring in Oregon, Washington, down into California, a little bit into Canada. And then the Rocky Mountain tail frogs. There's actually two species of tailed frogs in the Northwest, one in the Rocky Mountain range and one in the West. So here are sister species that appear to be responding to mountain building. So let's go and talk about some more mountains. So here's the United States, or North America rather, and you can see some mountains shown there. And we've talked already about the Rocky Mountains and the Cascade Mountains. And so we're going to get to the Sierra Nevada very quickly, and then go to some of the others, the Coast Range and the Klamath-Siskiyou. But I want you to follow my direction as I show, look at the Rocky Mountains and follow it down. And you can see a right parentheses that comes into New Mexico down there. So that mountain range at the very tip there is called the Jemez Mountains. And there is a Jemez Mountain salamander that is a sister species to the large mountain salamander that occurs in Oregon and Washington. And so here is a range map of the large mountain salamander in Oregon and Washington, and then this Jemez Mountain salamander that occurs in New Mexico. They are sister species, very highly related. So they are separated due to mountain building. And I think in this case, we have the Sierra Nevada coming into play and the rain shadow that it caused in Nevada. And then the Cascade Range, of course, and the rain shadow of the high desert in Oregon and Eastern Washington. So there's a story, there's a geologic story developing of salamanders. These are not highly mobile species. They need moisture. And you just don't see them trekking along the landscape very often, maybe during a rainstorm. So finally, we're going to do one more set of mountain ranges. And so you can see in this map, I've doctored it. There's a yellow circle and this giant H next to it. So the mountain ranges essentially represent an H in the Pacific Northwest going down into California. You have the Coast Range along the coast and the Cascades and Sierra Nevada inland. And the crossbar of the H are the Klamath-Siskiyous. If you know about a writer named Wallace, David Rains Wallace, he wrote a book about the Klamath Knot. And essentially the Klamath Knot is the crossbar of this H. It's where the intersection of all these mountain ranges occurs, and it's a highly diverse, biodiverse area. The plants are very different in these areas, as well as the invertebrates and the amphibians. And so at the Klamath Knot, there are more stories going on of amphibian biodiversity. So on the left, you can see some timeline if you're interested in those time frames. The Klamath-Siskiyou Mountains are actually pretty old. They were developing even at the time of the split of Pangaea. The Coast Range is pretty young. 30 to 60 million years ago. So there is a time frame of how these different mountains were being built, but the net result is that this Klamath Knot area is very interesting, and new species are being detected there almost every year. So I got interested in the Klamath Knot because of my work with the U.S. Forest Service. And so on the leftmost map, you will see the federal lands that are moist coniferous forests in our region. And those federal lands became part of the Federal Northwest Forest Plan, which was aimed to sustain species with old forest associations. The blue outline of that map on the left is the range of the northern spotted owl. And the northern spotted owl was a late successional and old growth associated species that was trending towards listing in the early 1990s. Analyses were done. Well, what other old forest species are in these moist coniferous forests? And there was quite a list of amphibians. As they developed the plan, the Northwest Forest Plan, they put reserves aside, they put riparian areas aside. What species fell out of that and were not protected by reserves or riparian areas? And there were 5 rare, little-known terrestrial salamanders that may have been of concern at that time. And we've talked about some of these already. So the ones on the right, we have already talked about 1, 2, and 3. Those are the large mountain salamander, the Shasta salamander, and the Van Dyke salamander. So now we're going to add in the 2 others that we haven't talked about, and those appear, their ranges occur right at that California-Oregon border. And if you remember, that's at the Klamath Knot. So here's the Klamath Knot story for those. If you look at the range map on the right, there's some yellow dots and some green dots. The green dots represent the Del Norte salamander, and the yellow dots represent the Siskiyou Mountain salamander. The range of the Siskiyou Mountain salamander with the yellow dots is smaller. So we're going to start with that one because it has an interesting story. In 1993, at the beginning of the Northwest Forest Plan, we only knew of 47 sites of the Siskiyou Mountain salamander. And it was put into a program called Survey and Manage. Let's survey for it, and if we find it, we will manage that site for persistence because there aren't very many of these sites. This is a rare and little-known animal. We developed a program called Strategic Surveys because we wanted to grow our understanding of the ecology of this organism. So we started surveying the periphery of its range to see where the range really was and could we expand our knowledge? Did it have a bigger footprint at the Klamath Knot, for example? And as each new location was found, I asked the field crews to please take some tissue from each animal. And for salamanders, it's really easy to take a tail tip. And we would use that tissue to do genetic analyses to see what was happening for genetic biodiversity of these animals. So these salamanders are so tiny, we could not take a toe. A toe is too small, but we can take a tail tip, and that was big enough to do the genetic work. The outcome of that is that we revealed a new species. And also at the end of the day, today, for example, there are over 500 sites of the Siskiyou Mountain salamander. So we really grew our knowledge. And today it's not considered so rare and little known anymore. But we'll get to that in a moment. So here's that genetic work, and you might recognize, here's that phylogenetic tree again, although it's been tipped. So instead of with the prongs going up, the prongs go to the right. But it's the same idea. The basal lineage is the lines on the leftmost side of this image. And the basal organism was the new one found, and it was found in Scott River area of California. And that was a unique form that had never been discovered before. That is the Scott bar salamander today. It was described as a new species. And it was the ancestral ancestral lineage. And from it, the Siskiyou Mountain salamander and the Del Norte salamander was derived. First, the Del Norte salamander derived, and then from the Del Norte became the Siskiyou Mountain salamander. So I just think this genetic work is almost like magic. They can put molecular clocks on this divergence, and it was estimated that the Scott bar salamander diverged into these other species in an era called the Pliocene, about 2 to 5 million years ago. And furthermore, the Siskiyou Mountain salamander, which was the one we had started with only a handful of sites, actually, there are two significant populations that are divergent, but they are not considered different species at this time, but we call them population 1 and population 2. So let me put this into a map so I'll give you an idea of what we think happened. On the left is the new species that we found in California called the Scott bar salamander near the Scott River. That is where it occurs today, tiny little dot. From the Scott bar salamander, the Del Norte salamander was derived. The middle image shows today's range of the Del Norte salamander. From the Del Norte, the Siskiyou Mountain salamander was derived, and you can see the arrow pointing toward today's range of the Siskiyou Mountain salamander occurs, which is the range on the right. And the down arrow shows that population 2 was derived from population 1, and population 2 occurs primarily in California. So there's sort of the map of what we think happened. And so consistent with the Klamath Knot being an area of local endemism for a lot of different organisms, it is an area of divergence for salamanders as well. And here's a close-up of the Siskiyou Mountain salamander and what has happened over the years since the discovery of this genetic population difference, the top two circles, and then the Scott bar salamander is the lower circle on the right. The top circle, the northern population of the Siskiyou Mountain salamander, has been put into a tri-agency conservation strategy. U.S. Fish and Wildlife Service has bargained with BLM and the U.S. Forest Service and agreed to maintain the sustainability of the northern population through a conservation strategy. This conservation strategy addresses measures for timber harvest and for fire, and it allows fuel reduction in areas that appear to be at high risk for fire. So it integrates people's needs as well as the needs of the salamander. That happened in 2007. And then just 2 years ago, or 3 years ago now, the southern population of the Siskiyou Mountain salamander, which is the middle circle, and the bottom new species, those two have been put into a second conservation strategy with two agencies. And the two agencies are the Klamath National Forest of the Forest Service and U.S. Fish and Wildlife Service. They have agreed to maintain the sustainability of those populations, allowing for timber harvest and fuels reduction and fire management to address the threats that are important for people. So this is a win-win that we are maintaining these species as well as socioeconomic benefits for people in this area. So this is an example of how our greater understanding of the phylogenetics, the genetics of these organisms, the species that occur here, integrating geology into understanding why this has occurred, and conservation. So conservation genetics is more than just identifying species and populations. There's another whole arm of genetics called landscape genetics, which is a way to track how organisms move across the landscapes, tracking gene flow or dispersal of these animals. And landscape genetics has been applied to stream breeding amphibians quite a lot. And here's just a model of some streams The lines are streams, headwaters going into larger streams, and ideas of how amphibians associated with streams might move around the landscape are depicted by these various buffer areas or habitat between different drainages. And a few years ago, we did not really have a good idea of how stream breeding amphibians emerged out of streams and then moved around the landscape. But we know a lot more now, and our knowledge is growing as more and more of these landscape genetic studies are being applied. But the big question, could these stream breeders move readily between different watersheds? And we did analyses, and the answer is yes. And so, for example, the coastal giant salamander is shown in that big picture at the top. And using genetic analysis, landscape genetics, we're essentially are using circuit theory, how electricity might move across an environment, applying that to how these genetic relationships are shown across a landscape. Yes, there is gene flow across headwater ridgelines, across different watersheds for coastal giant salamanders. However, we also found in that study that there was a latitudinal gradient in genetic variation. Lower genetic variation of these salamanders to the north compared to the south. It appeared that they are still chasing glacial retreat from the end of the Pleistocene. And so, yes, these animals are moving around, but it's not fast. And so there is still a signature of past populations and moving in a northward direction. Torrent salamanders are another example of stream salamanders, and they also are moving across headwater ridgelines. So my final example is part of Chris Cousins' research with another type of torrent salamander, the Cascade torrent salamander. And this paper is now in press in the Journal of Biogeography. And I want you to look at the range on the left, generally Oregon, Washington, Cascades. The picture in the middle is very similar to what we have been seeing with other species, that there are distinct populations across the extent of a species range. And to the right, this image looks just like what we just saw for the Scott bar salamander. We have a basal lineage, which is the yellow one. It's to the north. And then we have derived lineages, which are the other color-coded populations. Oh, and then I'm reminded to tie this to geography or geology again. If you look along the Columbia River, which is splitting Oregon and Washington, you see a few populations that are half blue and half red. So the solid blue dots are in Oregon and the red dots are in Washington. But this is the location of the Bridge of the Gods. And so there is a signature of landslides, or when that geography was connected and the river did not flow. Also, between the blue dots at the south and the yellow or orange dots at Mount Hood, there's a space. And that represents a location where we think the Columbia River used to flow in the Pliocene. So I'm finishing up now. The landscape genomics work that Chris Cousins is doing is unfinished at this time, but here is just an example of one watershed, and he's finding distinct populations within a single watershed. His investigation is, well, what metrics are these, is dispersal related to? And he doesn't have the answer to that yet. So in summary, thank you for listening to my story of geology and amphibians and genetics and conservation. Some of the take-home points is that we're still learning, and the ghost of past geologic events is evident in today's amphibian occurrences. And we have some ways that this is informing conservation today. And we're interested in pursuing this further, especially the dispersal aspects of how are the different habitats and populations connected, and is that going to be a new conservation um, sort of focus into the future. So I'd like to thank you all for being here, Chris Cousins for his work, and our colleagues and families. Thank you.
Oh, thank you so much, Dede. That was fascinating. All right. Well, we're going to take a few minutes, or Suze is going to take a few minutes. Suze Wehr from the Western Environmental Law Center is going to take a few minutes to talk to us about an exciting opportunity to do some conservation here in Oregon. And just as a reminder, if folks have questions, please use the little Q&A bubble and we'll get to those in a little bit. We may end up going a few minutes over 7 o'clock, but you can always come back and watch the recording if you didn't hear your question answered. But Suze, I'll turn it over to you.
All right. Hey, everybody. Suze Wehr here. And first of all, Dede, thank you so much for taking us on that journey with your presentation. That was so cool. And yeah, y'all, just for context, Dede is one of my science heroes. She was so willing to answer all of my amphibian questions while I was working on a project last year and is just a brilliant, kind human. So it's just a treat to hear from you. And now I'm here to talk about a way that we can help our amphibian friends in a huge way right now by passing 1.25% for wildlife, aka HB 4134. To get things started, the thing you need to know about amphibians is that they are indicator species. So when they're in trouble, they're sensing the things that are happening in the ecosystem before anyone else. And when they're in trouble, it's kind of spelling trouble for everyone. And right now, 18 out of the 31 native Oregon amphibians are on the list of species requiring urgent conservation action. And yikes, first of all. But likewise, 320 species broadly are on this list as well, and 11 strategy habitats are now at risk. All of this comes from the 2025 update to our state wildlife action plan. So this is going on, and amphibians are showing us what's happening, but only 2% of the Oregon Department of Fish and Wildlife's budget currently goes to wildlife conservation. And that may come as a surprise. You may think of Oregon as a really green state and figure that our taxes go to conservation, right? Unfortunately, like not really, not very much. Only a tiny amount of general funds actually goes to ODFW's budget, and it has to be allocated to really specific projects. Right now, there's like a hunting access one and a climate related one, but they're not working on conservation, or ODFW is, but those funds aren't. And the reason for this is really due to the North American model of wildlife conservation, which was built to manage game species and to prioritize consumptive species. So most of ODFW's budget, like wildlife agencies across the US, comes from the sale of hunting and fishing licenses and hunting and fishing equipment. And because of this, it's a huge issue in Oregon and in many, many other states, most states. There's sort of a bake sale approach to trying to raise conservation specific dollars and for non-game species, especially, where wildlife agencies will use fundraisers or special stamp designs or license plates that you may have seen to kind of, get whatever scattered funds that they can. But it's really not enough. And with 88% of the species in Oregon being non-game, there's a serious number of creatures being left out of the picture. But the states that have been able to establish enough funding to go towards wildlife conservation and actually support non-game species, what they've done is establish a long-term significant amount of funding dedicated to that, specific area. Missouri does it with a percentage of their sales tax. Virginia does it with a percentage of the sales of outdoor like equipment, outdoor recreation equipment. And we don't do sales tax here in Oregon, but we do have the third lowest statewide tourism tax. It's called the transient lodging tax, or I'll refer to it as the TLT. And this was identified actually years ago as one of the best solutions for creating a new funding source. It's deeply connected to our natural resources. Tourism depends on our natural resources. It'd be really cool if it could also help fund it through this TLT. It's a great solution also because it's not a cost of living increase for Oregonians. And especially as we're seeing federal threats increase with, threats to the Endangered Species Act happening, National Environmental Policy Act under attack, states need to step up as soon as possible, and Oregon included. So how would 1.25% work? Well, and hopefully you can see the screen. I don't know if my videos are blocking it. But basically, it would raise our statewide tourism tax from 1.25% to 2.25%. By doing so, we would remain the third lowest TLT in the US. And this is not a tax on businesses. This is not harming small businesses. It applies to the consumer when they basically pay for short-term rentals like hotels or Airbnbs. But that also means that the majority of it is paid for by visitors to our state. I think it's 65%. It's definitely over 60, is paid for by visitors who often come to Oregon for our beautiful natural resources and ecosystems. And other really cool thing is that in addition to finally getting to implement our state wildlife action plan, which is ODFW's like, hey, here's our key conservation priorities, here's the species, here's the habitats we need to focus on. In addition to actually funding that, this bill will also be able to put funding towards anti-poaching efforts and invasive species management and even habitat connectivity, like the beautiful wildlife crossings that save lives across the state, and you can kind of see in the illustration here. There is, however, a winding road ahead of us in this session. Even though it's a short session, we still got a lot of steps to go through. The bill was introduced to the House, and it just went into the Revenue Committee for its first hearing, which happened today. It'll go to the House floor for a vote, and then basically do the same thing on the other side with the Senate, and then hopefully the governor will sign it into law. And the really cool thing is that even though there's all these steps that have to happen, there is action that you can take at every single step to support it along the way, and we need your voice. And yeah, sometimes this policy stuff can be always really complicated, and the stuff is changing all the time, and it's a lot of hard work, but these good laws that we can establish really make our care real for the nature we say that we love. It's about making just like acting out justice, with our care for these creatures and places. And there's a lot of different ways to take action, which I'm excited to share. And I won't go into a ton of detail on this because I know Oregon Wild will be sending out info afterwards. But these are, some of these are 5-minute actions. Some of these are in-person advocacy that you can do, but there's a wide variety of skill sets, and wherever you are, like, you can jump into this. You can supply written testimony for committee hearings, like we had earlier today. It matters so much. And just as an example, over 1,000 people submitted written testimony for the hearing today, over 90% of which was in support. And legislators can't ignore that. That is a really big deal. And if we can continue to show that support each stage that the bill progresses to, that will help so much, to say the least. So keep that up. Another thing I'll highlight real quick is our Wildlife Day of Action that is coming up on the 11th next week. Technically, the last day to RSVP was on the 2nd, and that was mostly to confirm t-shirts or like lobby meetings, but you can still very much join us in Salem. There's going to be an amazing rally, and we're just all going to come together and show legislators how much this bill means to us and how much we believe in how it will help Oregon for years to come. Yeah, so there's other ways to take action as well, but check out the info that Oregon Wild shares. And one more note, these sessions go by so quickly. Like, they're called short sessions, but they're so short. So I would just recommend making a plan, signing up for updates from orgs like Oregon Wild, and just know that every action you're taking is really, really important. And I am so excited to be a part of this campaign and to have seen all the progress that we've already made so far. And the goal is we get this passed, and then hopefully we all get to take a nap like these giant salamanders here, because it's a long process. So yeah, if you want to check out any more of my illustration or writing work, I have my guidebook link here in my Substack, and I'm just, yeah, so excited to share with you all. And I think we need to make history by passing HB 4134. Thanks.
Awesome. Thank you, Suze. your illustrations really bring the politics to life because they're so cute. So thank you for that. Okay, well, thank you both so much for honestly tying all of that together. And we're gonna go for maybe 5 or so minutes over. Folks can hang out with us and answer a few questions. I've got a couple about the bill, the wildlife bill, but I'm going to save those because I think I can probably answer some of those in our follow-up email, or if we get to them in a minute. But a few about your presentation, Dede. One interesting one is someone asked whether amphibians were quicker adapters than other species or like categories of species like mammals or insects or reptiles. I thought that was interesting. Any idea?
Um, I do not know that they are quicker adapters. My experience is that they may have narrower habitats that they're adapted to. So they require moisture, for example, and cool places. And there can be little pockets of these habitats, little refugia, and those refugia can be isolated from other areas. So animals can find these refugia and then be geographically separated from other animals of its species, and then they diverge. And so it's not that they have the ability to change faster, I don't think. I think that they may get stuck places, and then they just either they drift apart or they adapt to the local conditions. And this was seen in particular. There's new work coming out of California and Nevada. For example, in Nevada, there are a lot of isolated wetlands, and the toads, the Anaxarus toads, western toads, are being described as new species in a lot of these isolated wetlands. And so it's applying the new genetic analytical techniques to something that they always knew. Oh, there's toads over in this isolated place, but now they can look at it and they're identifying new species. And the same thing in California. There are isolated wet places in this arid California landscape, and as they look, there's like, oh, there's a salamander here, and it's different. So I don't think it's faster. I think it's just possibly remnant habitats are isolated.
Makes a lot of sense. Yeah. Someone was interested in the limestone salamanders that you brought up that there are also some in Italy and wondered if there are any in the Oregon caves, thinking about caves.
So I recently went to Oregon Caves, and I saw a little brochure, and there is a skeleton of a salamander from Oregon Caves, but it had not been identified to what it was at the time. This was like, I think 2 years ago. So they have the fossil, but they, as far as I know, have not done genetics. I think they can't. There is the ability to do genetics on that, but the answer today is no. However, that author I mentioned, Wallace, wrote a book about finding a new salamander species in that area, and I have no doubt that that may come to light sometime. It could be there and we just don't know.
Yeah, makes sense. I guess I mean, this isn't a question, but from the audience, but I guess that kind of brings up, some of the issue around needing funding so that people can actually go out and look, right?
Yeah.
Using all the various techniques of sampling that you talked about, Dede. Somebody did ask a question about like how do we even identify, habitat like ephemeral ponds, right? Like I think a lot of the answer is people actually have to go out and look, and that
Yeah.
takes funding for research. Um, which
Yeah. When there's money for research, amphibians and also reptiles lack social capital. The money would go to birds and mammals oftentimes first, and people aren't so interested in that research on amphibians and reptiles. They just aren't as sexy or whatever it is that it takes. But That has been a prolonged struggle for a lot of researchers to find money to do the work. And of course, the pendulum swings on that topic, and there will be future times when there is more funding for that. But there is, like I said, we're in a race to discover what species we have before they're gone. And that's becoming more and more recognized. And so that is an effort that a lot of people are working on across a lot of different taxonomic groups, and especially some of the invertebrates are very little known.
Hopefully, after seeing this presentation, at least the folks that were here today will have more appreciation for the miracle of these animals and where they live and how they got here. I think I'll try to leave it there. Although, Suze, if you have any last things to say about HB 4134 before we go. Okay. Well, thank you both so much again, and we'll be sending some follow-up information on those ways to take action and a recording to this webcast. And again, thank you both so much, and we'll see you again soon. Good night.
Thank you.